So welcome to the latest episode of the Oxford Brain Diagnostics guest speaker series. A few words about Oxford Brain Diagnostics. We're a university spin out from Oxford University with a unique method for analyzing brain MRI based on cellular structure. We have an FDA cleared software tool that provides unique insights into patients' brains that is available for clinicians to use. And we also use our CDM analysis to support drug developers in their clinical trials. And we are joined today by Dr. Henne Holstege, Associate Professor at the Amsterdam University Medical Center and Head of Section for the Genomics of Neurodegenerative Diseases and Aging. She is a member of the Amsterdam Alzheimer's Center and is also affiliated with the Technical University Delft. And Henne, I've extracted this information from the web. I hope you don't mind me reading it back to you, but you've dedicated your career to understanding the genetic mechanisms behind neurodegenerative diseases and cognitive resilience in the oldest old. And your groundbreaking work on cognitively healthy centenarians explores the biological factors that protect against age-related cognitive decline. And obviously, in sort of bringing this right up to speed with the potential clinical use, this could pave the way for preventative strategies against Alzheimer's and other neurodegenerative diseases. So there's the amazing potential for how we might innovate in new ways of treating or preventing these diseases. Henne, you've previously given plenary talks at both ADPD and AAIC, so we're delighted that we have the honor of having you here talking just to us today. And you're going to speak to us, I hope, about some of the latest updates from your 100+ study of centenarians.
Yeah, so basically the idea is to understand what we can learn from cognitively healthy centenarians. I'm actually, since November 1st, I'm a full professor also at the VIB KU Leuven. So that I spend half my time in Leuven and half my time in Amsterdam, because we want to do a lot of omics measurements on the centenarian brains, and I'll come back to that in my talk. So this is my source of inspiration. I'm going to close you so that I can see my own slides. Okay. This is Mrs. Hendrikje van Andel Schipper. She was born in 1890 and she died in 2005. And at the time of her death, I was actually doing my PhD in breast cancer. But when this happened, this was for me such an enormous event, because the fact that she died wasn't so interesting, but she died at 115 when she died, and she was cognitively completely healthy. And this for me, and I was still doing my PhD, but I thought, this is so interesting. I want to learn more about this. And the beautiful thing was that she decided to donate her body to science when she was 82 years old. So we had access to different parts of her body, and including her brain, which allowed us to see, okay, what is going on inside that brain that would allow somebody to be extremely old and not demented. And she also showed that by achieving this, it is possible for us humans to reach extreme ages without cognitive decline. And that for me was something so extraordinary that I thought I would really like to know more about this. So this is these are the Braak stages. I'm sure you're well aware that this is for tau, neurofibrillary tangles, which start in the hippocampus, but with increasing severity of dementia, you see that the entire cortex gets infested with tau tangles. And the interesting thing is that we also see this happening with individuals who are actually not demented, but that stage very limited. And this is Mrs. van Hendrikje van Andel's brain. This is her brain, and you see a little bit of tau here and also some tau here. However, this is not the disease kind of tau, because as you can see here, the disease kind of tau is in a different place in the hippocampus than this is here. And this is called primary age-related tau. So although she has, these disease-related tauopathies or maybe neuropathologies, it is not the disease-associated tauopathy or neuropathology. And so for some reason, she has been able to, for 115 years, to not accumulate these age-related taus and these disease-related taus. So this is purely age-related. So I should say so. So she did accumulate some age-related tau, but this was said Braak stage 2. But again, this is not the disease-related tau. And she also had no beta amyloid plaques, She had no vascular pathology in her brain, and she had also no atherosclerosis throughout her body. Now, what about her made her so special that this was actually possible? And for me, this was the reason to just leave breast cancer behind and full focus on this, because I wanted to know what is going on here. So again, she shows by herself that it is possible to reach extreme ages without any signs of cognitive decline. And now we want to find out how this is possible. So Mrs. Hendrikje van Andel-Schipper is, of course, only one individual, but we wanted to find out, okay, who else should we compare her with to identify, you know, this extraordinary way of maintaining cognitive health, but who do you compare her to? Who fits that description? And to do that, we looked at the birth generation in 1912. So we started the study in 2013. So at that point, everybody who was born in 1912 was 100 years old. So we decided to see, okay, what happened to all the babies that were born in 1912? So we're going to look at their mortality statistics and their dementia incidence. And so what you can see here is that this is the age, and here you can see the incidence mortality, and you can see that people who were born in 1912, there were 170,000 babies born in the Netherlands in 1912, and you can see that 10% did not reach their first year. And you can see that this mortality statistic dies. This is the log scale. It's very important. So this is very low deaths. But still here, this is 10%, and it dies really quickly. And then you can see a very small peak here. This was the Spanish flu that was there in the Netherlands also here in 1918. And then you can see that there's a small death rate here, which has to do with drowning or maybe car accidents or things like that. And then you can see here in 1940, the incidence, the start of the World War, in which specifically men, there's a small peak. And then in 1945, we had the Hunger Winter when men and women, there's a small peak in mortality. But then something really interesting happens. You can see here that then after 40 years of age, you see the effects of natural decline start to be visible in the mortality statistics. And it's actually a pretty nice straight line, which actually starts at 100 at 0 years old. But then, of course, you can't really see it because of other deaths being more prevalent. But this starts to become very interesting, where you can see that the mortality starts to increase in the log scale. And this is something that was known as the Gompertz law. And Gompertz in 1825 showed that aging is the cumulative loss of the capacity to oppose damage to the body, leading to an increase in mortality in the log scale. And basically what he says is that, you know, when you're young, you have the ability to oppose damage, for example, a cut or maybe something that hits you, and you can actually process that damage pretty well and then just fix it. But that possibility to do that becomes more and more difficult with increasing age. And that then leads to diseases, and in the end, of course, to mortality in the log scale. And this is not just true for humans, this is true for many animals. So then when we look at this, we see here that the centenarians are 100 years or older, and there are now 1,000 centenarians left from these individuals who were born in 1912. Now the mortality rate is 40%. So that means that every person who turns 100 years old, 40% will not reach 101. And then still in the log scale, this mortality rate increases. So so less and less individuals will reach another year. So that also means that there is very much a difference between individuals who, for example, who are 50 and who will reach 51. That chance is extremely high. But there's a complete difference in being 100 years and the ability to reach 101, because there is so much more things going on in the body that will not allow, for example, if you have a fall or maybe an infection, will not allow the body to recover from that. So what you can also see is that most individuals who reach 100 years old are female. And you can see here, there's an enormous gender mortality gap here in between 40 and let's say 100 years old. And that has for a large part to do with the smoking epidemic that we had in the Netherlands, but also in other European countries, where especially men started to smoke when they were about 15 years old, and they started to smoke nonstop. And this led to a huge mortality gender gap between men and women, which leads for a large part to this difference in men and women who are centenarians. Now, I must say that about 30% of the gender mortality gap is based on biological reasons, for which we still don't clearly know how, but 70% of this mortality gender gap is based on smoking. So what we can do now is we can say, okay, what happens to these individuals in terms of dementia? So now we put here the dementia incidence in this same graph, and you can see that it kind of starts to become prevalent after 60 years of age. But then when you use the Gompertz law, you can actually extrapolate this. So let's do that. Here we have the extrapolation of dementia incidence, and here we have the extrapolation of mortality. And here you can see that just right after 100 years old, the chance to get dementia is higher than the chance to die. So that means that somewhere after 100 years old, there should be no individuals left who are not demented. And when you do the math using the Gompertz curve, you see that this is after 100, you see that that is about after 105 years old, there should be nobody left who is not demented. Now, that's not true, because we know that Mrs. van der Schipper was 115 years old and she was not demented. So that means that somewhere after 100 years, there should be a deceleration of the incidence of dementia. And that means that somewhere after 100 years, there is an enrichment of individuals who are specifically protected against dementia. And so they may be enriched with genetic factors, or maybe they have a better lifestyle. But the individuals who reach 100 years and who are not demented, there must be something about them that is worthwhile to more closely investigate. And that's why we decided to set up the 100+ study, because cognitively healthy centenarians beat so much risk to get Alzheimer's disease. They must be protected somehow. And we want to know what makes them different, what makes them stand out like this tulip in a tulip field. So we decided to set up the 100+ study in Amsterdam. And so since 2013, so it's now June, we have 505 centenarians included, and we'd like to continue including centenarians until we have 1,000 individuals included. We can already see a lot in these individuals, which I'll discuss. But of course, especially for the genetics, which we do see is important, we'd like to continue including centenarians. So what we do is we visit the centenarians at their house. Basically, we see online, we Google. Is there a question? I heard something. Okay, so when a person turns 100 years old, then then the mayor from wherever they live goes to visit them with flowers and the photographer. And when the person is really well, this gets published in the local newspaper, and that's what we Google for. And so actually we're kind of lucky, because if a person is really demented, that usually doesn't happen. And I should say that by far the majority of centenarians are demented. So this group of individuals who is not demented is very special, and they are then usually publications are there in these local newspapers, and that's what we Google for. We send them a letter explaining who we are, what we do, and we ask them if they would be interested in joining our study. Now, not everybody's interested, because sometimes they are very fatigued, or sometimes they are, cognitively not so healthy after all. So we get this really selected group of individuals who are older than 100 and not demented, who we visit, and we go by car, we visit them at their house. And this first visit takes place where we have all sorts of questionnaires. We ask them about, did they drink, did they smoke? We ask them to prepare a pedigree for us so that we know exactly how old siblings were and parents. We also ask them to prepare a sibling from their spouse, a pedigree from their spouse, so that we know, whether there is an enrichment of longevity in the family of the centenarian. We ask them how many children have they had, where have they lived, what kind of occupations have they had, all sorts of things. And then we also take a blood sample. Then we go back about 1 week later to do neuropsychological tests. And we do this because then, first of all, in this first visit, they can be sometimes a little bit, they find it a little scary, this person from Amsterdam coming by, what will it be like? But then they know us a week later, and then we can actually test a well-rested brain so that we can do a very wide array of neuropsychological tests. And one of the things that we also ask is whether centenarians would be open or would be able to consider post-mortem brain donation, because we would like to also see what does the brain look like on the inside. And we go back every year to follow up to see who stays cognitively healthy and who cognitively declines, and the same for the physical changes. But when they do agree to brain donation, we go back every half year so that when the brain does come in, we know exactly how it functioned very shortly before death. So these are individuals in our study. And what I always love to do during talks, I always ask the audience, can you have a look at these individuals? And imagine you would run into somebody like this somewhere on the street. How old would you then think that these individuals are? And of course, there's no audience now, so I'll just say what I always hear. I always hear that they're about between 80 and 83, maybe 85, but between like the first part of 80s. But then when I show how old they really are, then they're almost 20 years younger than what most people think they would be if they would see them on the street. And we see that what is going on outside, this is Mrs. Hendrikje van Andel-Schipper, by the way, is also going on the inside. So what do? We go, and for the first visit, we have an interview, and we also ask for a general practitioner's report, so we know exactly which medications they're taking, which diseases they have been diagnosed with. We know a life and a medical history. Here's the pedigree that we ask for. So we do a neuropsychological test assessment, and we do this longitudinally. We ask for brain donations, so that we can look in neuropathology and all sorts of omics on the brain material. In some instances, we've asked centenarians to come into the hospital for an MRI. We also collect blood, and we don't just collect blood from the centenarians, but also from their siblings, also from their offspring, and also from the partners of their offspring. And we do this because we want to look in the immune system. We want to look at immunological studies. We have iPSCs generated. We want to look at hormones, blood cell counts, cytokines, and we also look at blood-based biomarkers. And then we also look at the genetics. We see that, so we have array or genotyping, we have short read and also long read sequencing. And we also collect feces to investigate the gut microbiome. So let's look at the interview. What do we see? So just a quick introduction to who they are. Centenarians are lucid when we include them. They come from a relatively high social economic status. They are higher educated than the individuals from their own birth cohort, and this is true for both men and women. They have slightly more children than the individuals from their own birth cohorts, and this might be due to their higher economic status. We see, when we look at smoking, we see that men also smoked, but a little bit less than the people from their own birth cohort. With regard to drinking, we see, or alcohol consumption, we see no differences between the individuals from their birth cohort. And we thought that we would be finding individuals who are very thin because of the caloric restriction idea, but this is definitely not the reason why centenarians in this group became older or reached this phenotype, because they are mostly not the thinnest individuals, but they are almost always very optimistic. And this is something that is true for centenarians across the globe, but we also found this so, I mean, this is a great fun to visit these centenarians, because they're always optimistic. They're always making jokes about things. They always look at the bright side of things. And when you talk to their children, you really see that this is the way they live their lives, always looking at or always finding something positive about every situation. And obviously this makes this, this part makes it a fun thing to visit these individuals. So now we're going to look at some neuropsychological assessments. So what we've done is we investigate the mini mental state examination next to many other tests. But what we see is that individuals who have this, so this is the MMSE here, the scores. Individuals who have a higher MMSE score are more likely to survive another year, and this is the survival of the general centenarians in the Dutch population. And you see that these individuals with the higher MMSEs have a lot larger or lot higher survival rate. So what we can see is that if you have an MMSE specifically higher than 26, they live longer. And when you have cognitive health at 100 years old, and then I mean an MMSE higher than 26 at 100 years old, it is likely that individuals like that will maintain that all the way until death. Not all, but I think about 70% will maintain that high cognitive performance until death. We do see that centenarians are a bit slower in their understanding. And we see that when we compare the clock drawing test with what we see in the brain, we see that that is sensitive for amyloid pathology. And this is the clock drawing test. And maybe you have seen this many times, but when we ask individuals to draw a clock and put the hands at 10 after 11, and this is what a centenarian might do, this is what an Alzheimer patient might do. And we see that being able to do this is extremely sensitive for there being amyloid in the brain. So when we take all of these different neuropsychological tests, so these are the domains that we test: memory, fluency, executive functioning, attention, and global cognition. We see that there is a variability here. And this for us is fantastic, because this variability allows us to correlate whatever we find in blood-based biomarkers or maybe in the brain. It allows us to compare this with the cognitive performance. And this allows us to say something about, okay, what can we measure that really correlates with cognitive performance, right? So the brain is donated shortly after this, so that we can really correlate these two variables. Right, so variation in cognitive performance allows association with molecular measures. So what can we learn? So today I'd like to discuss the neuropathology and the genetics. So basically, do we see resistance or do we see resilience to amyloid accumulation? And are centenarians genetically protected against Alzheimer's disease or not? So we're going to look at the brain donations. And one thing I think is interesting, oh yeah, so these are the individuals in my lab who have done this. Jeroen Hozemans is now at Roche, and Andrea Gans is now has already gotten her PhD, and so has Mon. So these are alumni, but these are still in the lab doing the actual analysis in this. And what we see when we look at the brains from the Netherlands Brain Bank, what we see that a 50-year-old individual based on neuropathology is not the same as, let's say, a 90-year-old individual. We see that these are all Alzheimer's disease patients, but we see that Alzheimer's disease patients at a young age are really dependent on amyloid and tau, so NFTs, neurofibrillary tangles. And at this age, you don't really see. So this is really depending on these two neuropathologies. And then there's not much of these other pathologies available or prevalent yet. But with increasing age, you see that this completely changes. You see that a 90-year-old individual dies, these are old brains, so people who have died with a lot less amyloid or tau, but with much more atherosclerosis and TDP-43 and infarct. That means that a brain from a 90-year-old Alzheimer's disease patient is very different from a brain from, let's say, a 55-year-old Alzheimer's disease patient. And I think we need to take that into account, because with increasing age, you see that these things do accumulate. And now the question is, to what extent is that really disease-related, and to what extent is that age-related? So these are the brains that we now have. Actually, we now have 125 brains. You see that most of them are female. This is the age at death. And so most centenarians who we included mostly at 100 years old, we see that they usually die between 102 and, let's say, 104, most of them, or 101 and 104. but there's also some individuals who become much older. So we have all this follow-up from these data, so we can really follow who stays healthy and who declines. And the thing is that some people do decline, and so that means that this brain collection is actually representative of the first steps that you see with cognitive decline, because we include them at 100 when they're all cognitively healthy, but then some decline and some maintain health. So this brain cohort is representative of the initial molecular changes leading to cognitive decline. And we have now also the data that we can use to associate that. So this is the neuropathology that we see in the centenarians, and I'm not going to go by them one by one, but what I want to show is that they're not all Mrs. Hendrikje van Andel schippers. This is Mrs. Hendrikje van Andel, who had no amyloid, who had a Braak stage 2, who had no atherosclerosis. But you can see that most people have a higher amyloid stage, have a higher Braak stage. Some people do have a neuritic plaques, and most people, almost all, have atherosclerosis of some sort. So this means that, first of all, Mrs. Hendrikje van Andel is extremely special. Maybe that's also the reason why she reached 115 years old. But that there is lots of things that we can correlate with, because most people do have different sorts of neuropathology in their brain when they die after 100 years. So now let's look at the tau tangles and the amyloid plaques. And then the question is, are they resilient to it or are they resistant to it? So first look at the amyloid plaques. So what we did is we took the middle frontal gyrus and the temporal lobe and the occipital lobe and the inferior parietal lobe to see what is the abundance of amyloid in this in the brain, and does that associate with cognitive performance? So this is what we did. We investigated the quantitative cortical A beta load in 95 centenarians. So basically, we took a bit of the cortex here, stained with amyloid, and you can see these stains here. And then we calculated which percentage of this part of the cortex is positive for amyloid, and that is usually, let's say, between 2 and 5%. That is amyloid positive. So when we take this and we compare it to Alzheimer's disease patients, then this is what we see. Here's 27 Alzheimer's disease patients, and you can see the redder, the more red it is, the higher the load. And you can see that it starts really at the prefrontal cortex and then goes to parietal, temporal, and occipital. And that there really is this trend where it starts at the frontal lobe and goes all the way to the back. And then when we look at the centenarians, this is what we see. These are the 95 centenarians, and we see that there's this part that looks just like Alzheimer's disease patients. And we see that there are centenarians, about 2/3 of them, that have very little amyloid in the brain. So then we can ask the question, are these centenarians resistant to the accumulation of amyloid? And are these resilient to the accumulation of amyloid? Now we can test this because we know exactly how these centenarians functioned right before death. Because if they are affected, then probably their cognitive performance is lower. So let's see. And this is what we see when we look at the amyloid load here. And these are the staging systems. So the Thal phase and the CERAD-NP score, CAA stage, type 1, type 2. But this is the amyloid load. And what we can see here is that especially the digit span forwards and the clock drawing test, which I showed before, is sensitive to having this higher level of amyloid. And the digit span backwards and the clock drawing test are both tests that associate with executive functioning. So basically what we see is that if you perform not so well on the clock, then it is likely that you have a higher A beta load in all of these cortical areas in the brain. So this goes to show that actually that our testing of the cognitive performance is sensible. And what we are doing currently now is that we're looking into the proteomics, and we're also associating that with how the brain functions, and this is actually working. So what we can now see is that the healthiest centenarians, they resisted the accumulation of amyloid. And those who declined somewhat were probably resilient in the beginning, or resistant in the beginning, but then they did start to accumulate amyloid after we included them in the study. Now, this is the clock drawing test. You can score 0 to 5 on this. And so these are the individuals who have low amyloid load, and these are the individuals who have high amyloid load. And you can see that the individuals with high amyloid load score lower on the clock drawing test. Now, these are 4 individuals who have high amyloid, but do really well on the clock drawing test. Now, we looked into these, and what we saw is that they have very low tau compared to what you would think for this high amyloid load. So so these individuals would be resilient to high amyloid, but resistant to accumulating tau afterwards. And that is now really what we see. We see that the centenarians in our cohort, basically, we see a variable amyloid load, but the tau, they just never go to that inflection point where they start to accumulate tau. So even though sometimes they have extremely high levels of amyloid. So then what makes these centenarians different? So the problem with centenarians is that, what do you compare them with? This has been a problem from the beginning of our study, because you want to compare centenarians with normal people, but those who fit that description is pretty difficult to define. So what we decided to do now is that we have an age continuum of individuals with Alzheimer's disease and non-demented individuals, and then the centenarians. And we are now doing this the same way, but then for 500 individuals who have different types of neurodegenerative diseases, which we compare the centenarians with. That is ongoing at the moment. We have this large cohort together with different brain regions. That is something that we are now investing in. But this is let's say, well, I wouldn't call it a pilot study, but this is the first study that we did. We are now finalizing the paper, but what we can clearly see is that with increasing age, you see that, for example, microtubule-related proteins, they decrease. This is a This is a protein study. So this is the proteins in the temporal lobe that we tested with proteomics. And you see that the proteins associated with microtubules decrease with increasing age. But in the centenarians, they still have a very high level. In fact, a level that associates with, for example, 20, 25 years younger profiling. And that is kind of, overlapping with what you see on the outside of the individuals, where people thought, well, maybe they're about 25 years younger, or maybe they're like 80, 85. That is exactly what you see here. So basically what we see, centenarians are not just younger by the way they look on the outside, but also on the inside. And we see this with different types of molecules, actually. We've been going into this deeper and deeper now, and we see that, for example, proteins associated with beta oxidation, that is really associated with cognitive performance. I should have put that in here, but I can tell you that we can clearly see that better beta oxidation, better energy use by your mitochondria, for example, really associates with higher cognitive performance. Now the question is, how is genetics involved in this? So we're going to this. These are the individuals in my group who are involved in this, and I should say that because of my background in cancer, that's where my interest in the genetics actually comes from. And my initial research questions to start this was really, okay, so we see these centenarians, and we see that this phenotype sometimes occurs in more individuals per family, so that you have several brothers and sisters who all are older than 100 and not demented. So there must be something genetic in them. So can we not find this out? Why don't we know this yet? What is that? So that's where my interest in the genetics comes from. And so there's a pretty big team that's investigating this. And what we have done based on the pedigrees is that we calculated sibling relative risk, basically the heritability of reaching a certain age when having a centenarian as a sibling. And you can see that to reach 60 years old, or 70 years old, you're for about 20% dependent on your genetics, but that this increases with increasing age. So basically what we see here is that to reach 100 years old, you are dependent for about 60% on your genetics. And that means that well, basically genetics are important and you have to be lucky with your genome. More on that later. Okay, so we wanted to find out whether, so this is the Alzheimer's disease GWAS, I'm sure you're all aware. So these are genetic loci that are more prevalent in Alzheimer's disease patients than in controls. These are genetic loci. We really don't know what is in them, but we want to know, do centenarians also carry them? This, of course, is the evidence for this genetic locus to be differentially present in Alzheimer's disease patients compared to controls. So we wanted to know for all of these genetic loci, whether centenarians are differentially affected by them or carry a different frequency of them compared to normal, let's say, middle-aged individuals compared to Alzheimer's disease patients. So this is what Nico did in his work, and we're currently also doing this with PD, with FTD, with all sorts of different neurodegenerative diseases, and we see very similar signals. But what we see is that the centenarians have this, we see this trend where the centenarians are enriched with the protective allele, and then come the normal controls, and then come the Alzheimer's disease patients. So to go a little bit deeper into this, this is ApoE4, clearly a risk increasing locus, ApoE2, a risk decreasing locus, and we can see that Alzheimer's disease patients have a high prevalence of the ApoE4 risk allele, then come the normal controls, then come the healthy centenarians. So they have the lowest frequency of the risk increasing ApoE4 allele. And we see the vice versa for ApoE2, where centenarians are relatively enriched with the protective ApoE2 allele, where Alzheimer's disease patients are relatively depleted with the protective ApoE2 allele. So when we look at this further, we can see that these are the biggest differences where the centenarians really stand out in what makes them different in terms of what we see in the Alzheimer's disease GWAS. And again, we see similar signals in the other GWASs for neurodegenerative diseases. So basically what we can see is when we take a polygenic risk score that the centenarians have a low polygenic risk score compared to Alzheimer's disease patients. However, we cannot still pull them apart, which means that what we're testing are just markers for a genetic region. We do not know what is actually defining this risk or what is actually driving this risk. You can see the enormous effect of the ApoE4 allele, and that is because it's just very, very prevalent, relatively speaking. So it has a high effect size, ApoE4, and it's prevalent, which makes it a very interesting gene actually, because that is usually not, it really makes it stand out to other genes with high effect sizes, which are usually much rarer in the population. So to make a long story short, they have less risk increasing genetic factors, they have more protective genetic factors, and now we want to know which factors they are. So we go to this graph, and I'm going to put you down a little bit here. So here you can see that what we did is we took that difference that you see between the centenarians and normal controls, and we sorted that according to the largest difference. And here you see this big difference come in these three genes, the SORT1 gene, the TMEM106B gene, and the progranulin gene, and those are all genes associated with the endolysosomal system. So what the centenarians are now telling us is they're saying, well, your endolysosomal system better function, otherwise you do not become a cognitively healthy centenarian. And so we are now looking more deeply into, okay, what about these loci do we see that is explaining this difference? I'm not going to go into that today because I don't have enough time, but we see very interesting things in these regions that might explain this difference. Another gene that we see is the HLA gene. And of course, many people know this, but so this is Daniel Alvarez-Silvent, who has actually looked into this. We looked into different HLA alleles, and here there's basically one allele that is in linkage with each other, where you can see that previous research in the UK Biobank has identified different alleles that have associated with disease. And so we found 59 HLA alleles associated with 12 diseases. And of those 59 HLA alleles, we saw that 6 HLA alleles are significantly depleted in centenarians. And these are the 6 alleles. And so these are in linkage with each other, as you can see here. And then these are not in linkage, and these are 3 independent HLA regions. And what we can see is that with the HLA alleles associated with Alzheimer's disease are very much differentially present in centenarians. So they are depleted in centenarians. But there's also a very interesting effect where you can see that this is synergistic pleiotropy. So basically, HLA alleles associated with a higher risk for disease associate with a lower chance to become a centenarian. And that is true, for example, for the Alzheimer's disease HLA alleles. But what we also see is that sometimes we see an antagonistic pleiotropy, so that you have a lower risk for disease, but also a lower chance to become a centenarian. And that is true, for example, for asthma and for celiac disease. And I should have, yeah, so now this is overlapping, but also for autoimmune thyroid disorders and for multiple sclerosis. So the interesting thing might be that when you have diseases that have a late onset versus early onset, that if you have early onset diseases, that so HLA alleles associated with protection against autoimmune diseases at a younger age may lead to disease at older ages. And that's the reason why they may be depleted in centenarians. So you really see this antagonistic pleiotropy take place. So something might be protective at a young age, but risk increasing at an old age. So, and then the last gene, this is PLCG2. You may have heard of that. We see that centenarians are enriched with the protective variant in PLCG2, and this is actually a coding variant, whereas most of the other regions or genetic risk loci are represented by a SNP that is uncoding or not coding. This PLCG2 SNP is coding, and it was identified before by Rebecca Sims in 2017. And basically what it does, it encodes another version of the phospholipase C gamma 2 protein, which is associated with neuroinflammation because it is downstream of the TREM2 receptor. And interesting thing is that we found a 104-year-old individual who carried the PLCG2 protective variant. And we had her come in to the hospital and we did some brain scanning. You see that it is positive, but well, the thing is, but she's nevertheless, she drew a perfect clock, and she was homozygous for the ApoE4 allele. So we don't know whether or not the protective effect of the PLCG2 protective variant actually, allowed for this ApoE4 homozygous individuals to reach 104 years with, no cognitive decline, but it might have at least contributed to it. So this led us to look a little deeper in how is this possible that this ApoE4 homozygous individual actually exists. I can't go into too deeply because of time, but we published this paper of this PLCG2 genetic variant, and we found, the fun thing about the centenarian study is that we can go to their family members, we know them, so we also know who is the carriers. Their children, half of them will be carriers of this genetic variant. So we just phoned them and asked them, can we draw blood so that we can have fresh blood and test this? And we did this in collaboration with Jacques van Dongen from Leiden. And so basically we drove around the country and went back and got fresh blood samples from all of these family members. Again, centenarians are enriched with this really rare variant, so this is also something nice that we can actually do, so that we could test all of these different types of blood cells, peripheral blood cells, to see whether or not they function differently in the siblings who were carriers versus the siblings who were non-carriers. And we found that carriers had a slightly more responsive immune system. We saw for the B cells that the PLCG2 phosphorylation was slightly higher and that the calcium response was slightly increased. And for myeloid cells, we saw that they slightly were more effective in phagocytosis and a small increase in ROS production. But I should say that this difference was very small, and which is, I think, very much understandable or explainable, because if you have something as strongly selected for as the immune system that was selected for over millions of years of evolution, you can't change it very much without any detrimental effects. So only small effects in these centenarians will, contribute to a longer life, or maybe a more responsive or an advantageous immune system response. Now, we thought that perhaps this would also explain why the centenarians, when COVID broke out, were not really affected. I really thought that this was the end of the cohort, but it really wasn't. They all survived. And we thought that perhaps the PLCG2 effect might actually also explain that this happened. But when we looked into individuals who were just vaccinated for COVID, because this actually happened right then that we did this experiment, we saw absolutely no differences. So we don't think that because of the PLCG2 enrichment or the protective variant, that this is the reason why they reached this extreme age, or why they survived COVID. But that might be, maybe the HLA system might be involved, but clearly they were not affected by it. So what we're doing right now is, so I'm for half my time in Leuven, where I have this now where we're bringing all of these brain tissues, where we're now going to measure a very large brain set according to all of these different omics, and we're going to actually put this on a server where this is going to be shared. People can actually access this data when it's generated. So we'd like to share this data with with anybody who would like to access it. So this is where I would like to end. So the question, of course, is why do some individuals reach 100 years in cognitive health while others suffer from dementia around the age of 70? Well, we have seen that centenarians accumulate dementia-associated neuropathology, but less. And and what we basically see is that those who have really high cognitive performance, they resisted the buildup of amyloid. And in some instances, they do have amyloid, but then they're resistant to the buildup of tau. Centenarians are genetically protected against dementia, different types of dementias. We see that the immune system of the centenarians may be slightly more active, but we also see that centenarians are based on very different parts of a better immune system to reach their extreme age, and that they depend on a functional and a lysosomal system. So what we do know is that this is what their phenotype is, the aggregated effect, or the additive effect of many small effects. But those are effects that we can learn from. For example, there are efforts underway to understand the effect of the protective PLCG2 genetic variant, but we are actually seeing many other protective alleles that are also enriched in centenarians, and this is just the beginning in that sense. We are identifying now what might be driving this, and we see that basically when you look into the long-read sequencing, we see that the integration of transposable elements in the alleles that centenarians are enriched with, that might be differentially regulating the genes in the vicinity, that might explain why certain haplotypes in these alleles are actually protective versus risk increasing that are enriched in Alzheimer's disease patients. So what did the centenarians, I always love to show this slide. This is what the centenarians say. We see the centenarians every year, and every time we ask them, why do you think that you reach this extreme age? And they say all sorts of different things, and there's never something the same, which means that it just happened to them. It's not something that they actually actively sought for. It's something that overcomes them. Yeah, so nothing is the same. And this is my research group, who I really have to thank for this, because this is a group that has many different sorts of expertise, and together we put everything together. These are the centenarians themselves, who of course deserve the biggest thanks, and my sponsors. And with that, I'd like to end.
Wonderful.
Stop sharing.
Thank you. Yes, thanks very much, Henne. That was great. Thank you for a very thorough and accessible summary of all of the work that you've been doing across a whole range of different types of investigation, which is always a huge challenge and very impressive. I mean, I've got a couple of questions myself, but we've got a few minutes maybe for the rest of the team to step in. I'm going to just take advantage of the fact that I'm speaking first and ask. You showed an image because we're an MRI organization. That's a lot of our focus, and you explained that you have done some MRI and some PET. You showed an image of one example near the end. I wondered if you could comment on any other findings or general insights that you've got from any of the imaging.
Yeah, so, and I do see that I'm way over time. I am so sorry about that. This 52. I'm so sorry. But so what we saw, so this APOE44 individual was, of course, unique, so that we asked her to come in, and we did see some amyloid there. The others were amyloid negative, but you do see, when you look in the MRIs, you do see the effects of aging. So basically, they have less white matter, and the cortex has shrunken. So you do see the effects of aging. They are older brains, and when you take the MRIs and you ask, let's say, clinicians who are used to judging MRIs, they would say, though, this is an Alzheimer's disease patient, 85 years old. That's what they would think.
Really?
But clearly not. Yeah. So we've done an experiment actually lately to put this APOE44 individual into context, because what do you see? So we wanted to have this objective judgment of what people think about centenarians in general. 85-year-old Alzheimer's disease patients, but they're not. Yeah,
that is interesting. With specific atrophy and kind of Alzheimer's disease areas, or just a generalized sense that it looks shrunken, atrophied.
Yeah. So, but they, yeah. I mean, I can't say it any differently than that people that those who judge them thought they were Alzheimer's disease patients.
Yeah.
And the funny thing is, clearly they're not. So. Yeah.
Very good. Okay, so let me open the floor, as it were, to questions from the rest of the team.
Yeah, I'll go. Hi, Henne, that was a fantastic talk. Thank you so much. So I'm kind of interested in what information that you guys have gleaned can help with clinical trials, and I'm wondering how you would limit subject selection based on this. Are there protective genes that you would like to remove from the pool of candidates being tested for these drugs? Something like
that. So basically, so that is one of the objectives that I have as a researcher, is to basically perfect the polygenic risk score. As you could see, we could not pull centenarians apart from Alzheimer's disease patients, but these are actually the strongest differences, so that there should be the highest effect size, and still we can't pull them apart. Now, I think that that is because what we're measuring with GWAS is just markers. They just mark an area in the genome. But if you zoom into those areas, there's so much going on in there, and you can see, okay, so centenarians are indeed usually carrying the protective allele, but what makes that allele different? And when you look more closely, you see that individuals in the population, that there's not a risk increasing allele and just a risk decreasing or protective allele. There's many more alleles. And some of them actually have this integration of these allele elements or other transposable elements that differentially regulate that specific haplotype, or the genes in that specific haplotype. Now, if we would annotate every individual according to those alleles, then we could probably, have a much better idea of who is really running a high risk, which might be interesting for clinical trials, and who is not so. And then also we can say, that's my personal interest also, we see that some of these alleles are more vulnerable for the effects of aging, because when an element integrates in the genome over evolution, it will accumulate all sorts of CPG sites around it, so that this thing won't jump around. And that might actually change with increasing age. So perhaps individuals who have this integration of this element in one of these haplotypes might be much more vulnerable to the effects of aging compared to people who carry another haplotype. So, but that's completely dependent on how this thing regulates the genome in the vicinity. But we think that this is where the future should lie in terms of identifying who is at risk and who is not at risk before symptoms occur.
Lovely, thank you.
Great. I think Mario has a question. Go ahead, Mario.
Yeah, can you hear me?
Yes.
Thank you very much for the presentation. So, I have a curiosity. GRN sort 1 are no other frontotemporal lobar degeneration risk factor, right?
Can you repeat that, please?
So, GRN and sort 1 are genes involved in the frontotemporal lobar degeneration risk factors. So, do you think this is very interesting to me, because do you think these genes reflect resilience to a broader neurodegenerative pathology rather than AD-specific protection?
Absolutely. So because of that, I think actually TMEM106B is in the Alzheimer's disease GWAS because FTD is mingling in all sorts of different neuropathologies associated with diverse neurodegenerative diseases are mingling in with increasing age. Now, because in the latest GWAS, the UK Biobank, so by proxy, Alzheimer's disease patients were included. Now these things are starting to come up, but they might actually be reflective of other neurodegenerative diseases that are co-occurring in older individuals. And that's when TMEM106B came up, and also other, for example, TDP43. Also, you see, increase with age, something that is associated with FTD, right? Now, when we looked at the FTD GWAS, which actually just a new one came out, we also see that the centenarians in the common loci are very much in risk with the protective allele. When we look at the PD, we see the same thing. Not for all alleles. I think AD is clearly the strongest effect, but when you look at PD and FTD, you see similar things for where you can really see centenarians enriched than the normal individuals and then the diseased individuals. So based on that, we can say that they are really dependent on a specific mechanism to reach this large age without cognitive decline.
Thank you.
Henne, can we keep you for another 5 minutes, just for a couple more questions?
Yes,
is that okay?
Yes. Sure.
Great. Okay. Thank you. So who else has a question? Yeah, go ahead.
Hello. Sorry. You mentioned that you did MMSC and other kinds of, these kinds of psychological tests, and you mentioned optimism. Were there other kind of personality traits that came across, or did you do any kind of testing on personality traits that kind of shone through in this cohort?
So we did look at whether people were depressed with a depression scale, and
okay.
clearly we see that centenarians are not depressed. Now, I should say, of course, that if you have, let's say, a distribution of age at death in the population like this, we're basically sampling all the way here, right? So this is a really odd population. It really selected. This is completely biased. So this is really something that really describes this population. They are not depressed and they are optimistic. Maybe that's the same thing. So this is also the reason why I started to collect hormones. I really want to understand whether or not that is also genetic and whether that is a hormonal thing. I haven't been able to look into that, unfortunately. Because no time, but I really would love to look more closely into that.
Thank you.
I was wondering if you have any identical twins in your study and if their omics profiles look the same.
So I think we have one, but we haven't looked in that omics yet because they're still alive. So we have to wait. And I don't know if they're both brain donor. I think only one is a brain donor. Yeah, so we'll have to wait to get another twin in, I guess. Yeah, I don't know that answer to that question.
Okay.
I might dive in with another one then, partly because with the kind of our approach, the technology we use, cortical disarray measurement, is inspired by looking at the detailed the detailed fine structure of the cortex. So it's not really a measure of a protein deposition or some other kind of classic neuropathology marker. It's about the kind of health of the brain and the intrinsic, the sort of the structural integrity of the brain. And so, if you like, it's a metric that kind of spans all the way from, if you very healthy potentially, to increasing disease degradation. And we've considered looking at it around the issue of brain reserve, and this kind of these words that you've used about resilience and resistance, I find very, very interesting, very compelling, because I guess I was thinking you've talked about the ways in which the brains of these individuals, and in fact these individuals in general, seem healthy, look like 80-plus-year-olds, suggesting that they would have a kind of, in some respects, you might think that they'd have an odd kind of brain age kind of assessment. But what you just said earlier on was that people who do a visual read of the brain scan suggest that they look like they've got sort of aged brains that in fact maybe even look quite diseased in profile, just to sort of at the sort of volumetric level. So I guess I'm just playing around with the idea that we've got this idea of resilience. Potentially, we've got some sense of cognitive reserve and brain reserve. Do you have a sense of how these pieces of the jigsaw puzzle could fit together? Is there a picture emerging from your perspective?
Well, I don't know in terms of structure of the brain, but we do see in the proteomics, we can clearly see that there are that, for example, the mitochondria are much more active in those who have a higher cognitive performance. And, that makes complete sense, right? So, and that the lipidomics, the lipid structure, the lipid processing is clearly, much younger than you would expect for that age. So that's where really what we can see now with the proteomics that we did thus far, where we see the largest signals, and with the brain, with the folding of the maintained proteostasis the proteins associated with proteostasis are much higher in abundance than those individuals, let's say, who do with normal aging, let's say. So that's probably what they depend on. This is something, of course, that we're now looking further into. But I can't really comment on the structure, but more so on the proteomics and on the genetics that we really want to combine. And it's working, actually, because when we look at, for example, individuals who carry a specific haplotype, we see that they have higher levels of a specific protein than, for example, than the individuals who carry a protective haplotype. So that deconvolution of those genetic regions is really starting to work out, and what you see is actually having an effect on the different abundances of protein in the brain. So that's what we really want to continue.
Yeah. So it might be as if, say, brain age and biological age and the different features of that, they're not all going together, although clearly there's a correspondence, which I guess is what you're investigating. But just thinking about the brain structural findings, it's as if some aspects of the biological system are compensating for what looks like kind of gradual structural degradation, perhaps. Yeah.
Well, so I mean, so if they say that it looks like an 85-year-old Alzheimer's disease patient, well, clearly they're not 85. They're sometimes 20 years older. Yeah. So that means that the biological and indeed the chronological age diverges, but then still you do look at a very aged brain. It's not like it doesn't look like a 50-year-old brain or something. So they are affected by the effects of aging, but not so much. I mean, it goes slower. It is as if those genetic regions are just not so affected by aging as others might be. And that's what I think we're looking at, that they're just not as much affected by increasing age. And then they can extend whatever happens to normal individuals, let's say that's 80, that occurs to them when they're 100 or 105. And I think that's what we're looking at, that's the effect we're seeing. And that's also probably why they are enriched with these protective factors, genetic factors, I should say. Yeah.
Fascinating. Thank you, Henne. I think we should draw this to a close now, but thank you for giving us such an excellent survey of all of the work that you're doing, and clearly it's continuing on, and it sounds like you're going to double the sample size, I think. Is that what you said?
I hope so. Yeah, it's going to take some time, but I think it's well worth it.
Absolutely. Yeah. Yes. So best of luck with that, and we look forward to seeing you again. Thank you for your time today.
Thank you so much for having me. Okay. Bye-bye.